can be broadly defined as photosynthetic organisms that are not higher plants. So it's a very big and diverse group, and it really includes all these photosynthetic organisms that basically don't have leaves or roots. Like I said, they carry out photosynthesis. Um, they are very important to our ecosystem because they serve as the basis of the food chain. So these are small cells. They use the sun's, they harness the sun's energy to grow very rapidly. And then other organisms consume the algae, and those organisms in turn get eaten by larger and larger organisms. So they are very important for the food chain. And not only that, because they are photosynthetic microorganisms that grow almost anywhere there is water and sunshine, they provide lots of oxygen, and they actually take in a lot of carbon dioxide. So these algae that grow, when you hear about phytoplankton like growing in the ocean, that's a form of algae. And once again, extremely important for providing oxygen to our environment and actually taking CO2 out of the environment. However, sometimes if they grow too much and proliferate too much, they start dying off, and then you have a large biomass that's kind of mucking up your lake or whatever, and that in turn can actually consume oxygen and can kill fish that depend on the oxygen and those sorts of things. And then there are a certain group of these organisms that also produce toxins. So not all algae produce toxins, but some do. And that's really what we're talking about tonight. So just to provide you some examples of what algae can look like. So this is what I call green filamentous algae. It's growing on these rocks, but you can see that the water in this stream is very clear. So the algae is growing in large chains, but they're kind of attached to the rocks, and the water remains quite clear. Kelp in our oceans is also a form of algae. It's called brown algae. And even though it does kind of look like it's got some structures like leaves, it is truly an algae. Extremely important to our marine environments. It provides a hiding place for fishes. Lots of organisms eat kelp, so very important. And then this is another example of algae. So this photo was actually taken at Scootney Reservoir. Those of you that don't know where Scootney Reservoir is in North Franklin County near Connell. And it's a place where a lot of people go fishing. And we typically see algal blooms there in the summer. And this is a photo I took. And so this is a case where the algae has grown so fast and so rapidly that it's kind of overtaking the lake. And in this instance, it's causing could be causing a little harm because it's at this point, it's probably dying off and decaying and consuming some of the oxygen in the lake. Unfortunately, some of the algae up at Scootney does produce a toxin. I'll get into that a little more in the evening. But when you see a situation like this, I think most people would not swim in that. They probably wouldn't allow their dogs to go in that. So one good thing about this type of algae is that there's a visual clue. We know it's there. We know it's probably not good for us. However, a lot of times you can't determine whether a toxin is being produced just by looking at the bloom. So when this occurs at Scootney, we usually find out about it by somebody calling up to complain that they've seen something like this, or we send one of our staff members up there. They see this, we take a water sample, and we test for toxin, but kind of getting ahead of myself. Here is a red tide in a marine environment. So algae is, like I said before, is also found in marine environments. It can form these red tides, and these red tides can be very harmful to other organisms as well. And unfortunately, filter feeders like clams and oysters can accumulate those toxins, and then when you eat the clams or oysters, you can become very sick. Another thing that was on the news earlier this spring was that there were a lot of sea lions in California that were coming on shore and kind of spinning in circles and doing strange things. They were affected by a toxin that's produced by phytoplankton in the ocean. And this is a shot from the Columbia River. The kind of brownish material that you see growing on the rocks is another type of algae. And yet you can see that the water here is still very clear. So some forms of algae grow on rocks. It doesn't really make the water turbid or cloudy. So who are the bad guys? Like I said, not all algae produce toxins. There's kind of a specific group of algae that produce toxins, and they are called cyanobacteria. And they are true bacteria. For those of you that took college biology, the bacteria are the prokaryotes. They're the tiny little single-celled organisms. Eukaryotes are the types of cells that we have. Anyway, these cyanobacteria are true bacteria. So it gets a little confusing because algae is such a diverse group of organisms, and only some of them are these cyanobacteria. And it's just these cyanobacteria that produce these, what we call cyanotoxins, and that's what makes dogs and people ill. So historically at the health district, we did have a toxic algae program, but mostly we just investigate when people would call us. Like somebody would go up to Scootney and say, hey, there's a green algal bloom up in Scootney. Can you go test? And we would do that. So that's kind of our mode of operation. And I kind of delve into this a little already, but I'll go into some more detail here. But basically, somebody gives us a report. We go out, we take a water sample. We don't know if it's toxic or not yet. We send it into a lab, and we actually have a lab here that can do some testing as well. And the lab sends us the results. Are there toxins present? And there can be about 4 or 5 different cyanotoxins. In the Pacific Northwest, microcystins and anatoxins seem to be the most common toxins in this area. There are a couple of others, but those are the 2 I'm going to focus on tonight. And the State Department of Health have set thresholds. So if the toxin is above a certain concentration, they deem that hazardous to your health or risky. And so we follow this sort of outline. So if we get the results back then and it says, in the case of anatoxin, if the anatoxin is more than 1 microgram per liter, then we will go out and set up warning signs. Once again, if the toxin level exceeds a certain threshold, we will go out and mark the shoreline where we detected that toxin with a sign that looks like this. You might see other signs like this. These other signs are just kind of like, this is what algae looks like. This is what you should be aware of. But when you see this is a really heightened awareness. This means that we found toxins there, and it could be hazardous to your health if you consume water, if you have a dog that ingests the water, that sort of thing.
I'm just zooming out for you, Jim, so they can see.
And then once we post those signs, we keep testing the water then every week. And if once the toxin level drops down below the threshold for 2 consecutive weeks, then we remove the signs and test less frequently. In the Tri-City area, kind of getting ahead of myself. Anyway, once we have confirmed for a couple of weeks that the toxin levels have dropped down, then we remove the signs and just continue with our routine monitoring. In certain instances, if the toxin levels are extremely high, or if we have somebody report a human illness or a dog dying, if it warrants that situation, you might see this sign. And that's just, it's a very hazardous situation. We've detected very, very high concentrations of toxins, or we've known that a dog or a human has gotten ill in that area. And I just showed you the sign, so that's another. Okay. So, like I said, our typical procedure of operation is that we would, go out, sample for water. And we never saw algal blooms in the Columbia River. If you've been on the Columbia River, it's actually quite clear, clean river. There's flow to it. It's usually quite cold. But in 2021, we had a report of a dog dying not too long after being in the river. He took the dog to the vet, and that vet, was familiar with some of the symptoms of a toxic algae poisoning, and brought it to our attention and to the owner's attention that this very well could be an algal toxin poisoning. And on top of that, I think it was the same vet treated 2 other dogs that same weekend with the same symptoms, and both of those dogs had also been swimming in the Columbia River. So this was very alarming, and yet when we went down to the river, we did not see any sign of an algal bloom going on. We talked to the State Department of Health, and they said, well, it very well could be toxic algae. We just don't see it. So take water samples. So we took water samples, and we found very high levels of anatoxin A, which is a very, very potent toxin that these cyanobacteria will produce. At one point, we recorded a level of 67.2 micrograms of anatoxin. So this is like 60 times above the recreational level. And these were in locations where the dogs had reportedly gone. And yet, if you go down to Howard Amen, and that was one of the places that was reported, you see something like this. Matter of fact, this is actually a little more turbid or cloudy than I usually see it. I think because it was in a sandy area, it was a little more turbid. But in general, you don't see anything that looks like a real significant algal bloom. So there's no visual cue that something's going wrong here. And yet we found very high levels of toxins. And I was just, for emphasis, I mean, this is typically what we see. This is kind of what we were seeing in the Columbia. So this was a very big deal for many reasons. Like I said, there was no obvious bloom. That's usually what we're using to key in on, and we didn't see that. It was surprising that we found this in a clear river. A lot of times you see algae in stagnant bodies of water, like a lake or a pond that doesn't have good water flow through it. But usually when you have flow through something like a river, it's mixing the water, so the water temperature is kind of constant. And if you get a lot of growth in one area, the river will kind of push it down and sort of mix things up. So we were just kind of surprised to find it in a river system like this. And of course, the Tri-Cities has a lot of tourism, a lot of water recreation. So this is a major impact to our area. And then on top of that, all the 3 major cities get their drinking water from the Columbia River: Richland, Pasco, and Kennewick. I put 4 cities here because West Richland gets their water from a well. but if there was ever an issue with their well, there's an intertie, so they could conceivably get some water from Richland. So all the cities in this area are getting their drinking water from the Columbia River. And if there's toxins in the Columbia River, are we drinking toxins? And that was the concern. The quick answer is no, we're not drinking toxins because the water treatment takes care of the toxins, but definitely something to be concerned about. Anatoxin A is typically what we find in the Columbia River, although we will find some microcystins. It's a very, very potent neurotoxin. It can cause muscle twitching, reduced movement, labored breathing, loss of coordination, gasping, convulsions, and even death. There have been no known reported human deaths from anatoxin poisoning in North America. However, there have been instances where people have eaten seafood that's been contaminated with anatoxins. And this typically was in France. I don't know of too many cases occurring in the United States, but I think there have been some cases. And those cases were studied, and those patients, when they consumed the seafood that had the anatoxin in, reported difficulty focusing, double vision, impaired coordination, dizziness, tinnitus, and cramping in the legs and abdomen. So when we talk about these cyanotoxins, the units we use are micrograms per liter. So that's a concentration. Micrograms is a very small amount. A liter is, X amount of water. We can also use the term parts per billion interchangeably. They basically describe the same amount. And I mentioned this before. So for anatoxin in the state of Washington, the recreational threshold is 1 microgram per liter. And there also is a drinking water standard or threshold, and that is set at a lesser amount at 0.3 micrograms per liter. And I'll just say this about a lot of these cyanotoxins in general. They haven't been studied very well in animal models, so the toxicology, I would say, varies from state to state. If you look at different states, they'll have their thresholds set at different heights, and it's just a basis of what the epidemiologists in that state, when they read the reports about the toxicology, some think it's more severe than others. So anyway, you'll find sort of inconsistencies going from state to state, and sometimes that causes a bit of confusion. But these are our thresholds for anatoxin in the state of Washington. So in 2021, the State Department of Health, the Department of Ecology, the city water treatment folks, and us, almost left out BFHD. We weren't sure how to tackle this problem, and the best sort of scenario we came up from is to take water samples wherever there's a water intake or a water treatment plant. It just so happens that all cities have 2 different water intake areas. So we sample water at each one of those water intakes, and this is pre-treatment. So we call it raw water. So before it even gets into the water treatment plant, we're testing this water. And then on top of that, we've gone to 6 different recreation sites in the Tri-Cities, from Richland all the way to Findlay, and some are on the Benton County side of the river and some are on the Franklin County river. So we've got kind of a large span of river covered where the main population is. And once again, we take these samples from these 12 different locations, And then we sample it throughout the growing season of algae. And I would say the growing season of algae is from about May until the first part of November. And the peak's kind of in September. I'm jumping ahead of myself. I'll get to that in just a minute. But so anyhow, we decided we would take water samples from these locations. And if we had instances that the toxin amounts were above thresholds, we would put up signs in recreation areas. If we had exceeded some levels in the water intakes, then that would elevate it to another level where we would send those samples to a more sophisticated lab so they can be more sensitive and get a better quantitation at a very low level in terms of, that's very important when you're talking about drinking water, because I said that drinking water level is set at a very low concentration. So I just explained this, but basically we're trying to protect the public, and I'm talking about drinking water and recreation water, by taking these samples on a periodic basis. If we do find exceedances, we try and post signs on the shoreline where we find these exceedances. And in certain instances, if we think it's severe enough, we'll actually alert the media and say, hey, we think it's really bad right now. Get the word out. We've had a report. Let's say we've confirmed a couple of dogs have died. We need to get this out right away. We take those test results and we post them on our website, and I've provided the link here. And then, like I said, occasionally we'll do a media release if it's severe enough or what we think is bad enough. Initially in 2021, because this was so new to our area, we started a publicity campaign, and we made up a lot of signs, met with a lot of people, tried to get the word out, made presentations such as this. And so these are some materials we've come up with. Hopefully you've seen these in our community. We had some available here. They might have got all used up, I don't know. But so hopefully you'll see signs like this. And it kind of shows you the symptoms, what to do if you think your dog's, gotten the algae on their fur, that sort of thing. I showed a couple of these signs as well. So these are just sort of informative signs. I mean, this is what algae looks like, and what to do in case you've come in contact with that algae. And if it's severe enough, as I mentioned before, we'll try and put these warning signs up so people that are down by the river that haven't been to our website or talked to other people should know that that area is a hazard. So here is a kind of a trend of toxins in the Columbia throughout the summer. Sorry, the text is so small, but I'll describe it to you. So on the x-axis, that is the timeline. So on this end we have May. On this end we have November. And these are different sites, so that the colors represent different sites. And then the peak height is the antitoxin concentration. So if you look at May, May and June, there's kind of a baseline level, but it's barely detectable. Starting in usually the end of July and on to August, the antitoxin starts to creep up. And usually in mid-September is when we see the most toxins, and we believe that's because the flows in the river are lowest in the fall, and also the water temperatures are the highest that they will be all season long. And those are kind of good conditions for an algae. They grow better when the water temperature is warmer, and if there's less flow, that's causing them to disperse less so they can kind of grow in one area better. Whereas a high flow would be sort of flushing them downstream. So this is kind of typical of what you'll see during the summer. And this is from 2023. We saw significant levels in 2023. Unfortunately, we had 2 dogs die in 2023, and we also had another illness reported. And this is just kind of a snapshot of 2003. So I've indicated that in August there was a dog illness reported. In September, we actually had a dog death at Columbia Point. Start of October, we also had another dog death at Howard Amon. And you can see just some of the toxin levels. So, you know, at times it was quite high. It was a level that's almost at 60 there. So in 2021, across the nation, this similar situation played out in other large rivers. There were multiple rivers across the United States that had some sort of algal activity. Some dogs were dying. So the EPA definitely noticed that there was a trend. So they decided to do, starting in 2023, they decided to start a research project, and they picked 7 large rivers across the United States, and the Columbia River, right at Leslie Groves, was one of the sites they picked to study. I'm going to jump ahead here just real quick, because I want to talk about a situation. So early on, I talked about these algal blooms that are very obvious. We call those obvious algal blooms, they're planktonic. And planktonic just means that those algal cells are suspended in the water column. And typically they're pretty close to the surface because they're photosynthetic organisms. Anyway, that form of growth where you see all this cloudiness, that's called a planktonic bloom. There are other algae that will grow on rocks and other substrates like logs, sticks that you'll find in the water. Those don't form this cloudy turbidness, if that's not quite a word, but you get what I mean. In the Columbia, what we typically have are these benthic algae. So benthic just means they're growing on the bottom. And this photo right here, hard to tell, but that's actually, those rocks are underwater. So the water still is very, very clear. So this water doesn't get turbid or very cloudy like that. And anyway, we call this benthic algae. And benthic algae is the type of algae we find in the Columbia and in other large rivers across the United States. So the EPA saw this trend and they said, we need to study benthic algae because this is going on across the United States, and benthic algae had not been studied very well. So we did not know too much about it. So they decided they would go to these 7 large rivers across the United States and come up with methods for sampling benthics, because it's quite different. Your procedure would be quite different. When we go out and we look at these planktonic blooms, we just take a bottle of water, scoop it up, and we have our sample. If you go out here, where's the best place to sample? Do you just go over here and scoop some water, or do you scrape off this growth from these rocks and put it in your bottle? And if I was sampling here, would I sample this green stuff and this brown stuff, or maybe sample them separately? It's hard to say. So what the EPA decided to do, and others are involved as well, but they came to our area and they tried to figure out a methodology for best studying this. And the reason I bring this up is because they didn't necessarily go looking for the culprit in our area. And that's a key point, because at this point, we really don't know which benthics are producing the toxins. So they came here and kind of came up with this methodology of this is the best way to sample a benthic and so on and so forth. They came back last year and did some more sampling, which will be very helpful, but all of their data hasn't come in yet. So we haven't seen that data yet, but hopefully some of the data that comes in will be helpful in us to sort of piece apart what's going on in terms of which benthics are producing these toxins and that sort of thing. There's another government agency called the USGS, that stands for U.S. Geological Society or Service. And they look at other parameters just like water temperature, how cloudy the river is, the flow levels. They can actually test chlorophyll levels in the water. So they're looking at some of those things which can be helpful. And I think I touched upon all these other points. And like I said, searching for benthics or knowing which benthics are producing these toxins is a difficult task because, so here's some more examples of what benthic algae can look like. All three of those top photos are from the Columbia. I took all three of those photos. So you can see it looks quite different on whatever rock you might happen upon. So when you have growth on rocks, the scientific term for it is periphyton. And that just means it's a collection of organisms. It's not just algae. It can be algae, it can be cyanobacteria, it can be fungus, it can be some invertebrates like little creepy crawly insects. Did I mention fungus? It can be a lot of different things all growing together in this common community. That makes our job tough because if we find toxins in the water nearby this rock, let's say, which organism is producing the toxins? If we take a sample of that and look at it under a microscope, we might see 6 different organisms, and those organisms are difficult to identify. So, and then here you have 3 different, very different looking organisms. So here you have kind of a brown one that's kind of a film. Here you have a very hairy green one. That one's kind of a combination of brown and green. And you can see there are other rocks in the area that don't have any growing on it at all. These photographs down here were taken in California. California has a lot of rivers that also have this problem, and they've actually researched it more than we have. So they have a lot of good data that's coming out right now to help us understand what's going on here. One thing I want to point out here is that sometimes the periphyton will actually grow real thick, and then for whatever reason, let's say it's kind of late in the summer and it just starts dying off, or let's say the water drops 10 feet and what's in water now becomes out of water and it's 106 degrees out there and it fries the algae, and then the water comes back up. The algae got killed by that high temperature and now it's floating on the surface. And that's kind of what's shown here. And when this happens, we call that an algal mat. And that's extremely important because as the algae dies, it will start releasing more toxin. And as that mat floats down the stream, it can wash up on the shore. And if your dog's walking along the shoreline and happens upon some of these mats and actually ingests them, that dog is ingesting, it can be ingesting a very large quantity of antitoxin, and they can die very rapidly from ingesting those toxins. And, you know, there's just a variety of ways and just a variety of growth on these rocks that you can see. Yeah. And I kind of alluded to this already, but a lot of times when you take a sample from this type of situation and you look under the microscope at it, you see very distinctive cells, and a lot of times it's one type of algae. It's not a whole bunch of algae. Now that's not always the case, but usually you just see one or two types of algae, and it can usually be pretty easily identifiable by these structures. However, if I take some material off of this green portion here and look at it under the microscope, you see all these green filaments, you see other materials in there. If you scratch off some of this brown stuff, you see this big gamish, and you see multiple organisms. So just studying these benthics is quite complicated because you find a mix of organisms together. So it's difficult to say which one's producing the toxins. I mentioned the mats before, so those mats will peel off of the rocks and they'll float downstream. This is actually a photo I took. There used to be a dock at Howard Eamon Park, and a lot of this material used to collect on it. A lot of this material are algal mats. These brown pieces and green pieces, those are all parts of algal mats. I plucked one of these brown ones out, sent it to the lab. They homogenized it. They found 720 micrograms per liter, so 700 times above the recreational threshold. So if a dog ingested this, it would probably die very rapidly. And there again, when you look at this under the microscope, it's kind of difficult to identify the microbes or the algae that are in there. In some instances, you just see these masses of, it's just organic material. We call it mucilage. It's just mucus, and lots of things are just attaching to it. So this is a video. This is a video I took. These rocks were up by Priest Rapids Dam, so quite a ways upstream from Richland, so about 35 miles upstream of Richland from us here. But these are similar to the mats I've seen in Howard Eamon. And to go back, I did have these organisms identified. They're called Formidium, and Formidium is an antitoxin producer. So here we might have some evidence of at least one type of algae that produces antitoxins in the Columbia. And when I was up by Priest Rapids last fall, I saw these. And what do I have to do? And so this is how easily the material can lift off the rocks. So this just kind of shows you, if the river flow changes greatly, it can wash the material off of the rocks. If the water level drops, the algae can get heated up and die off. And this is what I think we should all be concerned about, is when we see a situation like this. So I'll show it once again. And you shouldn't be using your bare hands to do this. Probably a good idea to use a gloved hand. The toxins have been shown to penetrate into your skin, but they can cause a rash. I haven't had it happen to me, but I should be wearing gloves there. But I don't know for a fact, but I think this is Formidium, the cyanobacteria that we determined does produce antitoxins. So I have seen benthic algae that I think are Formidium that could potentially be producing antitoxin in the Columbia River. And these are just another, these are a group of cyanotoxin, or excuse me, a group of cyanobacteria that are known to produce anatoxin, Anabaena, nostoc, oscillatoria, formidium, also known as microcoleus. So these are ones that could potentially be in the Columbia River producing anatoxins, and there's probably more, but these are the most common ones. So this is just a nice snapshot, an aerial view of the Columbia River. Here's us in the Tri-Cities. Where I saw that formidium is way up there. And I mentioned earlier that I've received some money to do research on the Columbia. So these are some prospective research sites that I've chosen. And this is what I hope to accomplish. So I'm going to go out with my team and we're going to walk the shorelines in certain areas and look at the material growing on the rocks. We are then going to photo catalog a lot of these rocks. Anything that we see that we think is benthic algae growing on rocks, we'll take photographs of it. We'll also take samples of this, try to identify it under the microscope, also send in pieces of those to assess what cyanotoxins, if cyanotoxins are present, and if so, which ones. And then hopefully we'll come up with this catalog of photographs, and then we'll have some tools to start moving forward. Like, this is what the hazardous algae looks like. This is where it grows. This is where you should stay away from. So that's our goal. And California, as I mentioned before, is already doing this. And so this is kind of what I hope to achieve. They've gone out, they've identified these algae. These are what they look like. Of course, there's going to be variation. It's not going to be black and white, but they've identified some of these more microorganisms. They know what they look like under the microscope. And so that's what we hope to do. And when you do that then, and you go out and you survey the shoreline, especially if you know where algae is common, say for instance, we know this brown material is formidium. If we go out and we do kind of a survey of the shoreline, and we see about 10% coverage on these rocks, that might not be a very hazardous situation. However, if we go back a week or 2 weeks later, and we see 80% of coverage of this formidium, then this would be a very high risk situation, and we could alert people to stay away from that area, to not go into the water, what have you. I'm very close to the end. So thanks for hanging in there. So my take-home messages. Benthics are very complex. So as I mentioned, you know, it's this mesh, this kind of conglomerate of growth, and we have to tease it apart to kind of find out which ones are present and which ones are producing the toxins. It's difficult to determine who the culprit is, if you will. Grab water samples. I'm referring to the routine water monitoring that we do, is very helpful. It lets us know if there's toxins in the water and we can alert people. But, you know, it's very, it changes quite rapidly just depending on the flow of the river. So it's ever changing. But if we start to get a grasp on what these benthics look like, we can kind of put together a photo catalog and say, these are the bad guys. If you're out on the shoreline and see these, you probably want to avoid that area. Likewise, if we go out and we see it and we see that there's lots growing in a certain area, then hopefully we can sound the alarm and say, we're seeing a lot of this type of benthic. You should probably avoid that area. This is my humble opinion here. I don't want to scare anyone away from the Columbia River. I love the Columbia River. I spent most of my weekend on the Columbia River. I swim in it occasionally. I don't think it presents much of a health hazard to adults just jumping in and swimming. And the reason for that is most of us aren't ingesting a lot of water when we swim. And if we do, it's a very small amount. The toxins fairly dispersed in the water column. So I don't think it presents a very large risk to humans that are just recreating in the water, water skiing, jet skiing, fishing, that sort of thing. However, those mats that break off and are in the solid form and they float along the shoreline are loaded with toxins or can be loaded with toxins, and they present a very large health risk to your dogs. And if you have young children like toddlers that have a tendency to put things in their mouth, they could be in danger as well. So that's my spiel. I don't think that we should be afraid of the river. I just think we have to be cautious with our pets and young children along the shoreline when we see these mats present. And down in New Zealand, they've had this situation for quite some years, and they've done a lot of research as well. And I think we have a lot to learn from them. I was searching one day on the internet and I found this video, and this is a woman from New Zealand that's spearheading a lot of research down in New Zealand. And she gives a little talk in this video, and I think it's a nice educational talk because it covers a lot of what I just covered. And it has some nice footage of those mats that you'll see in the water. Ooh, maybe it's not going to work. Ah, it is. So I'll be posting all these the slide deck from my presentation, and I just included some more learning here, some articles that are very good scientific articles that kind of explain the benthic algae and other rivers in New Zealand and also the California area. This website that I've included is from the organization down in California that's done a lot of great research on the benthic algae. And then here I have some contacts of a couple of folks at EPA that's done some work here, some researchers at USGS. And I think I'll stop there and let people ask questions if they do. You have a microphone near you? Okay. You have a microphone near you? You push that button.
Hello, can you hear me now? Okay, I have a few questions, and this is typically, there's a group of dog lovers in this room that we take our dogs down to either Howard Amon or Leslie Groves. Leslie Groves primarily. I assume that there is a testing site at Leslie Groves, proximity closest to like the volleyball court area.
Yes, exactly. That's where.
Great. Okay, that answers that. How often? You said weekly, starting May through September?
On the second day of the month. It's twice a month. And I take a test on a month. Usually we have those results by the next day.
Really?
And sometimes we have to confirm those results, but we usually have the results for the next day. That can vary. We try and post it within a day or two of taking it. But in some instances where we have to confirm it, we might put something like we've detected levels here, but we haven't confirmed the exact concentration. When we have determined the exact concentration, then we put that information in there.
Okay, so to confirm twice a month, and typically within a number, if there is a positive result that comes back, any kind of positive, the signage would be up within 3-4 days type of thing?
Yeah, good question. I mean, once we do the test here, it has to be confirmed by another lab before we'll actually post signs. So there could be a couple of day lag. We test in our own lab here, but then we have to, our lab isn't accredited. The state has a very formal process of accrediting a lab to do a certain test. We're not accredited. We look at our test as kind of a screening, and if we detect X amount, then we send that sample to the state lab, and then they confirm it. And the reason why we do that is because we're taking 12 samples at each sampling period. If we were to send all those to the state, I mean, the testing is very expensive. And so if we were to send 12 samples to the state, plus it overwhelms them, they say like, we can't analyze 12 samples from you. So we do this pre-screening. And if we do the pre-screening, maybe only one of them's got some toxin in it, then we send that, they test it, they confirm it, and then the official signage goes up. You're welcome. Yeah, we're very unique. We're the only health district in the state. We're the only health district in the state that has this capability of sampling for toxins right now. So even though, and I feel very confident about our testing, but officially to post signs, the protocol is that it's a certified lab. So anyway, I think we're doing better than most health jurisdictions. It's just that we have to get that confirmation from a state lab that's accredited.
One other follow-up question. Okay, let's say we see a yellow sign that says caution down at Leslie Groves. Oh my God, keep the dogs out of the water. Is it a good assumption that, there's multiple points where you can take your animals in the water and let them cool off during these hot months? say you're half a mile up or down river, does one automatically assume just keep out period everywhere?
Well, that's a good question, and I think it boils down to what you feel, uh, what
We-
you right. Um, we take samples at multiple locations, and that's why, and we only post these signs where we find the high concentration. So if we've taken it from Pasco side of the river and we're not finding toxins, I I can't say that it's safe, but
Yeah.
yeah. I mean, I think that's safe if you really care for your animals and really key in on those mats. I mean, if you're seeing those mats or what you think are mats, you got to keep your animals away from them. Yeah, can you turn on that microphone there? Yep, just push the button and turn green.
For the online listeners.
Got it. So my question pertains to any water that comes in through the irrigation systems. Like if I have a pond back outside my yard that's constantly flowing, but it's fed with sprinkler water. So is there anything that we should look out for in terms of, is there any kind of filtration that's happening before it gets to that subsystem? Or is there any information that we have about water that gets there that we should look out for? Because
That's an excellent question. It's come up before. Unfortunately, we just don't have the manpower to test irrigation water. And algae can grow in irrigation water, and it's just one of those things that it's just difficult to monitor. And this is an interesting story. I started out this whole talk about finding toxic algae up at Scootney. Well, if you know about how the whole Columbia Basin project works, they take water out of the Columbia River at Grand Coulee Dam. It goes into Banks Lake. From Banks Lake, it goes into another lake. From Banks Lake, it goes to it goes into Moses Lake. And from Moses Lake, it goes into O'Sullivan. Then it goes into then it goes into Scootney. And all irrigation water doesn't always get all used up. And so when you have leftover irrigation water, it goes back into the source. So irrigation water is going back into the Columbia. And when we first detected toxins in the Columbia, I thought, aha, it's coming from Scootney because we know there's toxins in Scootney. But the toxins in Scootney are the microcystins, which is a different toxin. In the Columbia, we see anatoxins. So those algae aren't coming from Scootney. It's kind of what I was picturing in my mind because that water comes from Scootney and it eventually goes back into the Columbia. So, I mean, do I think that algae can be in those irrigation ditches? Yes, I do, but I think it's a very difficult thing. And there are many, many irrigation ditches. There's just not enough manpower to go through. And another reason why we don't is we look at it from a public exposure route, and people aren't supposed to be swimming in irrigation ditches. It can be used to irrigate crops. I realize that, but I think that's less of a hazard. Some of these toxins are very susceptible to sunlight. It's interesting because these organisms are photosynthesizing, that they're exposed to the sunlight all the time. But these toxins are actually metabolic molecules inside their cells. And when they get released, they're more susceptible to sunlight and UV light and drying out. And then they become, I shouldn't say they're never toxic after this happens, but a lot of these break down over time. So by the time it gets out of the irrigation canal, you put it in your garden, maybe it gets on some of your plants, but it dries out, it gets hit by sunlight. Another interesting point while I'm on this is that you would think, so in nature in general, a lot of toxins are produced to like kill other things. So take for instance, there's bacteria that live in the soil and they produce antibiotics that we use actually to kill other bacteria. Naturally occurring in the soil, a lot of those bacteria produce those antibiotics naturally to kill other bacteria in their neighborhood, and that confirms them a competitive advantage. Well, it does not appear that these cyanotoxins give a competitive advantage to the cyanobacteria. Scientists believe these toxins are part of their metabolism. These are very, very primitive organisms. They've been around. They're some of the most oldest organisms on our planet. They evolved in a very harsh environment. And it's believed that these molecules are part of their metabolism and they're not produced to kill other things. So it's not like they're releasing them into the body. Usually the only reason they get released into the environment is the algae is dying, the cell's breaking apart, and now these molecules are spreading. And that's why I think these mats are the most dangerous, because the algae's dying and it's this big mat of cells and it's releasing all these toxins. Yeah, one more question.
So if you had a small subsystem, like for instance, I have a water feature in my backyard that has 3 small ponds that get filled up continuously, that's fed by either a hose or from the sprinkler system. As long as that water is moving then, so it doesn't have a whole lot of chance to be exposed to direct sunlight more than probably about 30, 40 seconds at a time. But as long as that's getting fed in daily, what would your assessment be? Because I mean, it gets full of algae all the time. I'm constantly out there scraping it out. There's just no way to identify the specific type. But it's my dog's favorite water source in the whole world. That's kind of where I'm getting at.
Yeah, I mean, there are algicides that you can put in the water. I mean, copper, small amounts of copper can have an algicide effect. So there are things that you can add, but I don't know how that would affect your dog as well. I mean, if the copper concentration was high enough, it probably could affect itself. But there are things you can add to, I think, hinder microbial growth. Yeah.
So if you just want to take zero risk, is there a temperature, or I mean, would you pull your dog from the river like July 1st and let him back in November 1st? If you're like, I'm zero risk, or is there something like when the water reaches like 74 degrees that you can use?
I think looking at that trend, like that graph that I showed, starting in about middle of July, there's a good possibility you'll start seeing toxins.
Then about November 1st.
Yep.
Say, okay.
I did. If you had mentioned like if the algae is not like if you identify it, just clean water.
You didn't say what to do.
Yeah, or wipe it down with a towel.
Oh.
I'll just, so the question is how best to take care of a pet, a dog that's been swimming in water that you are concerned about algae being in. Yeah, just if you have fresh water, hose them down. If you have a towel, you can just wipe them down. Just make sure that they don't start licking their fur immediately. If you see any signs that you think that the dog might be struggling, I would take them to the vet immediately, because the statistics that I've seen right now for our area show that the fatality is about 70%.
Is this
source that you can call or go to find out what areas have high toxins or should be avoided?
Yeah, let me show you real quick. See? So this is our website. I think this is kind of hard to find. You got to scroll down, way down to here to water, toxic algae blooms. Is it showing up? Okay, so this is our website. Yes. This is where you'll see the current updates. And then if you scroll down here, this is where we sample at, and you can zoom in and out and click on these. So this, yeah, this, why isn't the concentration showing up? Anyway, you should be able to go in here. A lot of these won't have anything. If we don't detect anything, we don't post it. So you can go on this, but this is great to know. If you go elsewhere in the state, you can go scroll down to here, and this is a statewide map. So you could, like, let's say you're going to Seattle and you're going to go to Green Lake, and Green Lake's, I don't know, let's see, or you're going to some lake south of Olympia, you click on it here, and in Silver Lake right now, there are 27.4 micrograms per liter of microsystems. That's why it has a red dot, a red diamond. So this is a great source. The one thing I'm going to point out about this for our area is that, remember how I said that the lab has to confirm it? All of our negative results are never confirmed. So if you go on, so people have told me a time or two, they go to a site that they find here, like let's go on one of these X's here. What's neat about this too is you can go back in time and look. So, so this is at Two Rivers Park. So this is a place where we sample. And the sample date here was that's weird. Oh, I know what this is. Okay, I received a complaint from Two Rivers earlier this year. And this one, we didn't have our test set up. So I sent this directly to the state. That's why this one's showing up. Because what I was going to point out is that a lot of the state's data doesn't show up because, like I said, if we have negative results, they don't get reported to the state. So if I was to go on to another site, but this is one I sent directly to the state because somebody had complained. They had been out to Two Rivers Park. They had seen a lot of algae, and they asked me to go investigate. And I actually went out there. I did find lots of algae, but it wasn't toxic. I sent it to the lab. The lab did not confirm any toxins. So, I mean, so here's a good example. So no, not a good example. Let's get one of our routine sites here. Okay, sorry. A lot of our routine sites here, when you go and you look at the data, it'll say basically last sampled in July of last year. Well, that's a little misleading because we're sampling here. It's just not getting on to their website because it's a negative result. So, and I think that's an important point to make because in most instances, we're finding nothing. It's just not getting reported that we're finding nothing. We only report it when it's significant. Okay, yeah, you in the back. Can you grab these microphones?
Thank you.
So when you do a test and you do find it positive, how long after that is it? I mean, can the water change the very next day or, okay, so is there any way for us as, recreational users or whatever, do we just have to keep, for instance, I'm a user of irrigation district water and also of Snake River water as well as the Columbia. So, and I know you can't speak to all of that, but in a, in a general rule of thumb, if you see a posting on here that says, okay, on July 7th, we did see elevated levels. And then let's say July 11th, are we to assume that it is still a risk, even though there's no visual algae present?
Very, very hard to say. Very hard to say. Yes.
So in the interest of time, I'm not going to ask a question, but I'm going to put out a request. If it's possible to provide any links for trending. So for example, you gave 2023 results. Is given that temperatures have been rising as well as this is new to our area, is there, and I think you answered it, was your website, you could see trending over the years. The second request would be, you mentioned the thresholds across the United States. Is there, and that's the micrograms per liter on what's acceptable. Is there anywhere where we could see a comparison to states on what is acceptable? So Minnesota versus Washington acceptable levels, just to see the difference.
I would like to see that.
If you took the time to go to individual state sites, couldn't you almost create that yourself?
Yeah.
I mean, it'd be time consuming, but for those of us who are retired, we could probably do that.
Don't you get a post?
Well, I don't want to keep anybody here longer, but if you have more questions, feel free to stick around. I'll be glad.
Very much. Thank you.
Welcome, thanks for coming out.